Hi class, in this video we're going to look at how to analyze the number of lipid droplets per cell. So if you counted total number of lipid droplets per cell, this is how you're going to work up this data. You identify your groups, so it's very important that you label the graph well. I've had lots of students in the past, lots of groups, where they didn't label things appropriately each step of the way. And then they, had multiple people working on it, and they had mix-ups in how they were analyzing the data. And then ultimately they had to go back and redo their counts, which added an additional week to their workload. So we don't want that. So it's very important that you label things appropriately. So here I like over-label, right? So this is our acid group, and here's our negative control, meaning the ethanol. And if you had more groups, you would just have more groups, right? So depending on how many groups you had, each group gets a column. And then each row is labeled by the cell, right? So remember, it's lipid droplets per cell. So in this particular cell one on this particular slide of their data, they counted 223 lipid droplets in that one cell. And then they did cell 2, 3, all the way down through 15. And so this depends on how many cells you actually counted. They did quite a few, as you can see. And so looking at the number of cells, right? So then they're going to, and you labeled that on your merged image. So you can always go back and, like, if you, questioned your counts, or let's say I questioned a data, I could go back and look at your merged image. I could say, okay, so cell 1 on the alpha-linoleic acid slide is that cell. I could count it. And if I got a completely different result, or if one of your peers, maybe you could check each other. Maybe everybody could, count the same cell and do a cross-check. That's one way you could think about it. But anyways, so you could verify your results. So you notice we had 15 cells that we counted in this condition. We had 10 cells we counted in this condition, okay? So that's important to note. So down here in number of cells, I could write 15 and 10. Right? So that would be information that you can gather. If you had a lot of cells, if you ever wanted to use this, right, you could also do this a different way. So you can hit equal sign and then the function count. And I'm going to make this a little bit bigger here so that we can, I'm going to make this bigger so that we can actually see what all we're doing. Okay. All right, so equal sign, count, and then double click, and then you would just highlight the terms. And what it does is it counts the squares, not the values. It doesn't really do anything manipulation with the values, it just counts the number of squares for you. So if you had a really long data set ever, that would be a really quick way to do it. And you can do the same thing here with the control, hit equal sign, count. If you don't like using equal signs, if you're more comfortable clicking the insert function, you can do that as well. So you can double click on this, backspace, hit the function, and then you can find whatever you're looking for here. There's count right there, so you'd count, and then you could highlight it, with this going on here like this, and then hit equal, and then it does essentially the same thing. So that's one thing you need to do. So then what I've done is, this is your raw data right up here. So then we want to translate that to the data that we're actually going to graph. And notice I've changed the titles here of what we're talking about here, and that is for simplicity purposes. You want your titles that are eventually going to be on the graph to be very, very simple, so minimalist. So like this is a control. I wouldn't put a big long word there. Just control is fine. It's a negative control. We know that, and you might want to put that in your text. But for the graph, you want it very simple, and this is alpha-linoleic acid. So how are we going to do this? Well, first thing we're going to do is we want to calculate the average number of lipid droplets per cell. So how are you going to do that? Is equal sign, and then it's an average. And what we're calculating is the mean, technically speaking. So we want to calculate the mean for this condition. So average, and then you can highlight your samples here and then hit enter. And so that would be the average number of lipid droplets per cell under this condition, alpha-linoleic acid. And you do the same thing for control, equal sign, average. Once again, this is a mean. And then calculate that. Hit enter. You can already see that not even one lipid droplet on average in the control. So it's going to be very different, right? But let's look at the variability in the data. So standard deviation allows us to look at variation of the data. So to calculate standard deviation is equal sign STD, and I like this one that says STDEV. Double click on that, highlight your data. Remember, we are in the alpha linoleic acid condition. Hit enter. That would be your standard deviation. So you can see the actual, average number of lipid droplets plus minus standard deviation is about 245 plus minus 50, right? So there's a huge amount of variation there, right? I mean, plus minus 50 on either side of 245, which is kind of large. So let's look over here and look at the standard deviation for this, STDEV, highlight our data, hit enter, and you can see that we have a variability of about, essentially 1. So on average, right, we have about a little bit less than 1, but we have a variability of 1, and we can see that pretty clearly here in the data. The last thing we want to calculate is something called the standard error of the mean. I think in the other video I accidentally just called it standard error, but it's standard error of the mean. And so this is how you calculate it. Here I've got this value here. It is essentially the standard deviation divided by the square root of the number of samples here. And so this is pretty straightforward because we've got all this data already here in this chart. So we would do equal sign, and then we're going to highlight our standard deviation, which is this here, divided by the square root. And how you get square root here is SQRT. And then you need to double click on that function. And then the square root of what? Well, the number of samples. Here it's 15. And then I like to close parentheses, otherwise it might give you an error. And then hit enter. And so that is the standard error of the mean for that sample, for that average right there. And then you do the same thing over here, equal sign. And then we need to hit the standard deviation divided by SQRT. Double click on that, and then number of samples below, close parentheses, hit enter. And so this is actually what the error that we're going to graph in our graph here in a second. The last calculation that you need to make before you make your graph is something called a student's t-test. Student's t-test. A student t-test allows us to compare two different groups for significance. And since we only have two groups here, it's pretty straightforward. But if you had multiple groups here, you would just have another comparison over here. Let's say you had oleic acid as a positive control versus your control, and you would also then do oleic versus alpha linolenic. So this is if you had like a positive control. You might actually have 3 comparisons. So the key is you want to compare every group that exists. So every possible pairing you want to make and do what we call a student t-test on it. Now here's how you're going to calculate a student t-test, but let's look at what it means. So what you're going to get from a student t-test is it's going to calculate something called a p-value. And if the value that you obtain from your test is less than 0.05, we're going to interpret that as there is a significant difference between the two groups. Okay? So meaning the numbers, the values that we got are significantly different, those means. If the p-value that you get is equal to or greater than 0.05, we can state that there is no significant difference between those groups, right? So they're essentially the same, or statistically speaking, there's really no difference between them, even though you may think there is. So this is a way scientifically to show, that a comparison of groups. So since we don't have these two here, I just made them up. We're not going to do it. We're just going to show you the one that is here. So how we would do that is equal sign, and then T-T-E-S-T, t-test. Double click on that. Now for this, it says array 1. So what array 1 is your first group. So you're going to highlight your first group. Then, very important, comma. Then it says array 2. That's your other comparable group, which is this one right here. Comma. And then it asks you for the tail distribution. So for if you've never dealt with a tail distribution before, I'm just going to tell you what to do here. You can learn more about this in advanced statistics class. But this is what we call a two-tailed distribution. All right, because we're interested in both sides of the mean. And then it's asking you, is it a paired comparison of these two groups? 2 sample equal variance assumed or 2 sample unequal variance assumed? Well, we're assuming equal variance, so we're going to put a 2 there. So it's the array, comma, the other comparable group, comma, 2, comma, 2, close parentheses, enter. And so that gives you the p-value for that particular comparison. And what do we expect? Of course, we expect a significant difference here, but we have to follow the statistics. And is that less than 0.05? Yes. And so what I would maybe say is p-value And I would actually say is less than 0.05. And that way, if you have multiple groups here, and let's say you have something here, let's say you have a number of 0.4 or something like here for the t-test, and you're like, well, that p-value is not significant. So then you would say p-value is greater than 0.05. And so what we would say is there is a significant difference between these groups, right? Whereas here you would say no difference between the groups. And so this is kind of like, I'm already writing conclusion statements. It's kind of nice to have that here so that when you go to do your website, you already have the information you need here, right? So this is just extra information. Obviously, these is not actually in this data set here. So now what we're going to do is we're going to take all the information that we have here and make it into a graph. So to do that, we're going to highlight the data of interest. So we want to graph the means. So this is why I said earlier it's very important that you keep things simple here, right? And so we want to make sure we have the exact same, fonts too. So everything's the same now. And so now we highlight the data and the table headers there. And so we're going to go to Insert. And then I just like a simple, column right here, sort of graph. We're going to remove the chart title because it just takes up space and it's very novice. So we want to make something that looks expert-like. And so you can already see an issue that we have with this data that sometimes arises is, the scale is just so different, right? And so sometimes you can change the scale so you can actually see that there's actually something there so it doesn't look like you're hiding data. But most of the time you'll have some type of graph here too. It's very rarely that it's going to be this low. Their data just was, arbitrary low for some reason. So some of the things we want to do is you want to look at your actual axes titles, and I like this. It's very simple. I might change the font. I might change the black versus gray. I don't like gray. It typically doesn't show up very well. So you might end up changing that. You can always do that later. Pretty simple to do. Over here we want a chart title. It doesn't have anything. So what we do is we click add axis titles. And I'm going to get rid of the horizontal because remember we don't really need that. But we do want a vertical axis. So we can type in here and we can say that this is the average number of lipid droplets per cell. All right, so something simple like that is perfectly fine. And so that would be very appropriate. You can also, instead of average, if you mean number of lipid droplets, because it is a mean, we calculated the mean, right? I just noticed I misspelled something here. So that there's a couple different ways you can do that. I prefer average. I don't know why. I just look at it. I'm like, I got to change that. So, and then, you can mess with, the size and making it look appropriate for the image itself. So, I mean, there's a couple different ways you can do that going on there. The next thing you want to do is you want to add error bars. So I click on the bars, right? And then, so you want to make sure you have both sets highlighted. And this is really easy because we made this table, we have everything in order. And so what we want to do is we're going to go to plus here, error bars, and it throws in some stock error bars that have completely meaningless data. But what we want is error bars that make sense. So we're going to go over here, go to more options, error bars, more options, and then it's going to pull up over here how to format it. We're going to go to custom, specify value, and now we can give the actual standard error of the mean that we calculated. We have positive, which above the bar, above the average, and negative is the error bar below the average. Since they're the same, it's plus minus, we're going to click on one, and we're going to highlight the standard error of the mean here. We can highlight both of those because they're in order. And then the same thing with the negative. We can highlight both of those and hit OK. And what it's done, as you'll notice, is this one's so small it doesn't even show up. But this one, you can actually see it's the actual error bar there. Now, if we're having troubles with, graphing this, we can change, how this is set up. So we can change how these axes are. So what I've done here is I've manipulated the chart data. You can come over here to this particular, when you click on the axis, you can click on this. You can click on options, and you can change the maximum and minimum there to kind of, reinforce, if you wanted to put in even the negative, 25 there, you could, maybe show if there is some kind of thing below the graph here in terms of error. But right now you can see we've actually, showed the error bars here. So things you want to do also is change colors. So I clicked on the error bars here, and I really don't like how they're showing up as sort of this gray color. So I might come over here and change that color from gray to more of a black, so it stands out. And I'll change maybe the width to to 1, and that helps us see that we actually have data there too. And then I'm also going to change, so you can change how these appear. So right now it has too many things there, but we can double click on one of these bars, and we can change the outline to black to help it stand out. Probably go to 1 just to keep everything the same. I'll go to fill, and I can change the fill, to maybe more of a gray. Depending on the color scheme of my website, I might change that appropriately. But we can clearly see we're going to have differences here, in how things are going. You can delete these lines here. There's all kinds of things that I like to do to clarify the graph. Like I said, I would change these probably to more of a darker font, things like that you can do in here. So now we got it in more of a visible, pleasing kind of format. And so, and more professional looking. So the last thing, a couple of things we want to do still is we want to showcase the T-test that we did, the p-values. So to do that, we know we have a p-value down here of less than 0.05. And since we only have one group, it's really easy. Check out the video on area towards the end. I walk you through, if you have 3 or more groups, how to showcase that. So if you have 3 or more groups and you're doing just lipid droplet counts per cell, I'm not going to show that in this video, but you can check out the area analysis of area of lipid droplets. And at the very end of that video, I show you how to do what I'm about to do with just one comparison. I'm do it with multiple comparison groups. So if you have multiple comparison groups, you want to check out that. But if you just have one, it's really, really straightforward. And so what we're going to do is I'm going to create some type of character. I like asterisks. That's pretty standardized in the field. And so if it gives you that weird box, you would say no fill, no lines, because you want it to look professional. And so then what you want to do is insert a line. So to do that, we can come in and insert a line. And so you have to actually click on the table, but you can do format, and then I can bring in a line here. And we want to make sure it's level. I like to go from the center to the center. You can do it a couple different ways. And then we can change that to black. I like the width to be the same as their lines, just to be for consistency purposes. And so then we bring this down here, and that's an appropriate way to say that I'm comparing these two groups, and I found something important, something significant here. And so then your figure legend below this figure, you would say that the asterisk equals a p-value of less than 0.05. And then you would, very important the figure legends are. You can tell us what groups you're comparing. You can tell us that we've calculated, the graphs is showing the mean average number of lipid droplets per cell. You can tell us how many cells in each condition, right? So since we have different numbers of cells in each condition, always try to do the same. It always makes data analysis a lot better, right? So it would have been really easy for this group to maybe get a couple more cells so they're the same. So then you can just put it somewhere in the graph. You can say n is equal to 15 if they were the same. But because they're different, you kind of have to maybe in the figure legend, or you can even write below here, you can type in alpha linoleic acid, and you could even do something like n is equal to 15, or something like that. And I don't really like that because it makes things really long here. But there's different ways that you can showcase that. But come back to the p-value. If you don't like asterisks, you can actually, if there's space, right, if it's not too problematic, you can actually type in p-value is less than 0.05. Why not? You have the room in this huge graph to kind of do that, and then you could kind of center it, and then that looks really nice. So at this point, you have the means graphed, you have it appropriately labeled, you have something indicating p-value of the student t-test, and in the figure legend you actually say student t-test was performed, right? Otherwise they don't know what type of statistical analysis you did. And then we have the standard error of the mean graphed here, and you would actually label that in your figure legend as well. Standard error of the mean was calculated plus minus standard error of mean of the average number of lipid droplets per cell. And so that's all presented here in this graph. Very important that you save this graph. So you can copy it, right? You can copy that into like Paint and save it as a TIFF, save it as a JPEG, save it multiple ways. Also very important that you save all of this data. Save it a couple different locations. The last thing you want is to lose this data and have to redo it all. So save it at a couple different locations, okay, when you're completely done with it. And so that's kind of, this is what you're going to report then for your data analysis.